For the cell biology presentation, we have been given the topic hepatic stellate cell, which will be now known as HSC. The names of the group is Fergus McCormack, Eamon Kilgallen, and Aisling McDermott. I will now begin the presentation, starting with the introduction. The HSC, also known as the parasinusoidal cell, are parasites found in the parasinusoidal space of the liver, a small area between the blood vessels, sinusoids, and hepatocytes, also known as the space of DC. The stellate cell is a major cell involved in liver fibrosis, which is the formation of scar tissue in response to liver damage. The parasinusoidal space is a location between the liver between a hepatocyte and a sinusoid. It contains blood plasma microvilli of hepatocytes extend into this space, allowing proteins and other plasma components from sinusoids to be absorbed by the hepatocytes. The HSCs are star-shaped structures. They comprise approximately one-third of the non-parenchymal cell population and 15% of the total number of resident cells in normal liver. Now, the non-parenchymal exert both positive and negative influences on the hepatocyte proliferation, including provision of an extracellular matrix-bound reservoir of hepatocyte growth factor that is achieved after damage priming of hepatocytes for DNA synthesis through rapid generation of TNF alpha at later time points that curb hepatocytes DNA synthesis and initiate reconstruction and reformation of matrix proteins. Now, in this diagram, it shows a healthy liver versus an injured liver. Note the differences. In healthy liver, hepatic stellate cells are kept quiescent, which is dormant, and their main function is to store vitamin A droplets, which, when the liver is injured, hepatic stellate cells transform into activated myofibroblast-like cells to generate scar tissue. Now, as you can see, they are Kupffer cells. Now, a Kupffer cell is a specialized macrophages located in the liver, lining the walls of the sinusoids that form part of the mononuclear phagocyte system. There is also an endothelial cell. Now, the endothelium cell plays a central role, and active role in regulating the exchange of macromolecules, solutes, and fluid between the blood and the surrounding tissues. The high permeability of capillary endothelium are reflected in the presence of special transporting systems represented by vesicles, channels, diaphragms, and fenestrae, which in this case is fenestrae, as you can see by the healthy liver, and then the injured liver, we have a loss of fenestrae. Endothelial transport is a complex process in which substances are transported according to size, charge, and chemistry. The function of these cells play pivotal roles in the regulation of retinoid hemostasis. They house specific receptors of a retinoid binding protein. This is where vitamin A is mobilized from liver stores, and from there is transported in plasma in the form of a lipid alcohol retinol. Now, in this slide, it shows the activation of the HSC. Now, the activation of HSC is the dominant event in liver fibrosis. The early events in the organization of HSC activation has been termed initiation. Initiation encompasses rapid changes in gene expression and phenotype that render the cells responsive to cytokines and other local stimuli. Cellular responses following initiation are termed perpetuation, which encompasses those cellular events that amplify the activated phenotype through enhanced growth factor expression and responsiveness. Multiple cells and cytokines play a part in the regulation of HSC activation. HSC activation consists of discrete phenotype responses, mainly proliferation, contractility, fibrogenesis, matrix degeneration, chemostasis, and retinoid loss. Now on to the fun part, the fun facts. Research has found that HSC in Arctic animals such as polar bears and Arctic foxes store about 20 to 100 times the levels of retinoids found in human or rat populations. A study discovered that distinct populations of HSC in the mouse liver have different capacities for retinoid and lipid storage. HSC are the main matrix producing cells in the process of liver fibrosis, which was discussed in the activation of HSC. When HSCs were first discovered by a German scientist, Kupfer, which as you note in the diagram of the healthy and unhealthy liver, there actually is a cell called the Kupfer cell. So Kupfer in 1876, his initial description of Selete cells using a gold chloride method that identifies vitamin A containing droplets, referring to these cells as Sternzellen, which now the pronunciation might not be right, but it means star cells in German. Now the last slide shows our references. That is the end of the presentation. Thank you for listening. This presentation was done by Fergus McCormack, Emmet Gallen, and Aisling McDermott.